Since 1977, the Saba Lions Club has been a pillar of service and community care on the island. What began with 20 committed members has grown into a legacy of giving back, supporting those in need, and advancing the global mission of Lions Club International. From cultural events to signature service projects like Meals on Wheels and the Food Bank, the Saba Lions Club supports the elderly, disabled, and struggling families with compassion and consistency. Their health programs, like annual eye care support and glucose fairs, continue to promote wellness across the island. The club is also a champion for the environment, proudly maintaining an adopted hiking trail and backing local cleanup efforts to protect Saba's natural beauty. Several of its members have been honored with the prestigious Melvin Jones Fellow Award for their outstanding dedication, proof that the spirit of service is alive and well on Saba. The Saba Lions Club, serving with heart for the good of all.
Good evening, everyone. Good evening. Welcome to another night of See and Learn. Unfortunately, we were not able to enjoy the beautiful location at Island Flavor once again because of the weather, but we are grateful to Sunny Valley Youth Center for hosting us tonight. We'd also like to thank a few of our sponsors at the beginning. The Lions Club is responsible for the live streaming of this event, so not only do we have people in person, but we also have a virtual audience watching from Canada, the US, the Netherlands, other parts of Europe, wherever. So thank you to the Saba Lions Club. We'd also like to give a big round of applause for our main sponsors of See and Learn, without whose help this would not be possible. So we'd like to thank Public Entity Saba, the Nature and Environment Policy Plan, and the Culture Funds of the Caribbean, CCG. And a big round of applause to all of our sponsors. We have close to 60 all in total. So thank you to all of our sponsors. And tonight's expert is sponsored by Juliana's Hotel. So we'd like to thank Juliana's for hosting them. We'd also like to thank Island Flavor for sponsoring the dinner tonight. So thank you for supporting Island Flavor if you're sticking around for dinner. And we do have a raffle going on this month, in case you haven't heard this news already. We are selling raffle tickets for $5 a ticket, and the proceeds go towards funding our scientists who will be coming next January through March. Most of them are insect scientists, and they will be collecting baseline data for Saba. I have our little temperature thermometer here. And as you can see, our goal is $5,000 or 1,000 raffle tickets. And we are just at about the $1,800 mark. So there is plenty of room to buy raffle tickets still. Just saying. Upcoming activities at a glance. Tonight's speaker, is having a field project tomorrow where we're going to be doing some toxin testing at a few different ponds throughout Saba. So if you are interested in joining, please let Lynn or myself know. And we will be leaving the See and Learn tent in Windward Side at 3 o'clock. Then our next presentation is this Wednesday at Calibri Cafe with our sargassum expert, Vincent Encomio, who is right there. So Vincent will be our next speaker, and we look forward to that as well. And if you participate in one of our upcoming field projects, you receive a free bucket hat or a baseball cap. And they're made from recycled materials. They're super soft. They're very nice. So please participate in a field project. As an associate professor of medicinal chemistry at the University of Michigan, Dr. Tim Cernak takes tools from cutting-edge drug discovery and applies them to some of the planet's trickiest conservation puzzles, from tackling fibropapillomatosis in sea turtles to fighting chytrid fungus in frogs and even advocating for more sustainable biomedical practices. Tim is proving that saving species sometimes starts in the lab. Before joining the academic world, Tim spent nearly a decade at Merck designing life-saving medicines, and now he holds multiple research appointments across global health, chemistry, and data science. Whether it's invasive species or infectious diseases, his lab is all about using chemistry to react to real-world environmental challenges. Let's give a big round of applause for Dr. Tim Cernak.
Thank you. Thank you, Emily, so much for that wonderful introduction and for this invitation to come here. My God, I get to visit some really interesting places in this career. This one kind of takes the cake. So thank you all for coming out and sharing your body heat today, and you know, hopefully I can raise the temperature a little bit in here too. Right. So I'm on a journey to try to understand what I can do as a chemist in this space. As Emily described, I spent a decade working in pharma, both big pharma and in biotech, and I loved it. The work is incredibly rewarding. I still do work in this space to try to treat human diseases. But yet, as I looked around, I could see a lot of other health issues that were maybe a little harder to define, at least as a pharmaceutical chemist. But I had trained to be a pharmaceutical chemist. So, can we look at the world today and imagine how we would come to a health crisis with some of the modern tools that we have. We live in an incredible era of medicine right now, so I think it's a really interesting time to imagine this. And so a big part of what I'm doing here when I'm not preparing for this and doing other work is trying to write a complement to this paper. This paper is a real landmark for me. I was unaware of it, and I started to read and learn about it, and it's titled, What is Conservation Biology? And I think, you know, maybe today in 2025, maybe you have some notion about what conservation biology means to you. But in 1985, when this paper was published, it was very quixotic. It wasn't easy to define what is a conservation biologist, what can they do? And now as a chemist, as I'll describe, it's much harder to define what it is that a chemist can do, and I'll talk about that. But my hope is that by the end of the week, I'll be making a lot of polishing touches on a paper called What is Conservation Chemistry? One of the challenges is one, defining what needs to be done, right? So this is a figure from Michael Soule's landmark paper in 1985, and he tries to establish that conservation biology is, he calls this a synthetic discipline, and he calls it a crisis discipline, which I find is very, very interesting. And you know, it was known that conservation works by 1985, right? Pandas were coming back, humpback whales were coming back, bald eagles were coming back. We'll talk about that in a moment. But it wasn't clear how you would define how to do that. Like, how would you structure a conservation intervention? And it's not quite cancer biology, right? It has aspects of cancer biology that come into this, but it's different. Anyways, I get to hang out with a lot of conservation biologists in my career now, and it's wonderful. They teach me about natural resources. They're very big on genetic tools. But it's not clear what a chemist can do. And so I try to engage in this fight, but one of the things that's a big challenge for me is this. So every time I approach a conservation biologist and I say, what if a chemist can help? They say, yeah, but those chemicals killed bald eagles, right? So it is true that chemistry has a stain on its reputation, of having deployed DDT as one potential chemical. It's easy to villainize a chemist in the space of conservation. It's that like nothing is easier, right? And I am 100% on board with this being, you know, a major challenge. But I would say is that, you know, chemists do have things to bring to the table, and that's what I hope we can do, is that we can find perhaps more precision approaches to deploying chemicals. This is out the back of an airplane, and this is very much a broad spectrum insecticide. Not good, right? Meanwhile, here's where I got my start. And so we're going to go on a little detour through organic chemistry, and there's going to be no quiz at all. And if you loved organic chemistry, if you ever took it, then you'll be like a kid in a candy store. And if you hated organic chemistry, there's like 6 slides, and we'll get through them quick. But when I saw these types of molecules in my graduate career, I was like, that's what I want to do. I want to draw hexagons for the rest of my life. That's how I want to see the world. And all of these come from nature. They're various anti-cancer compounds. This one came from the Pacific yew tree. This one comes from vinca, little purple periwinkle plants. But anyways, this is my worldview. So I look at conservation challenges, and I want to see the hexagons inside. And I have enjoyed, as was described, a career in the pharmaceutical industry where we would imagine making such molecules to treat any type of medicine. I've worked in hypertension, I've worked in diabetes, cancer, infectious diseases, Alzheimer's, all with big pharma, and then in precision cancer with a biotech company. And we imagine that we're going to go through this cycle really quickly. We'll design molecules, we'll make them as a synthetic organic chemist. I try to make them. We like take our Lego building blocks and we click them together. And if we can make it, if you're lucky enough to make the chemical bond you aspire to make, you get to test it in some type of biochemical assay. What that looks like in a modern drug discovery lab is you've got some purified proteins from your patient's cancer, and you also have some patient cell lines. So routinely we would go to the clinic, we would get some cancer cell lines from an excised tumor, and we would grow them up inside of a well plate, a 96 well plate, like Beth and I were playing with earlier today. And then we would make new types of chemicals that maybe could thwart that cancer and test them. This is a multi-trillion-dollar industry at this point. There's, probably a lot of people have a favorite molecule that has been invented in this way. And I have enjoyed a career watching the front lines of this industry that can make these wonderful medicines. Here's just some of the work from my lab that we've gotten a lot of kudos for. We used AI to make this molecule very rapidly. This is impressive. If you are really into hardcore organic chemistry, you'll agree with me. If not, then we use robots, we use data science to try and navigate through these really big combinatorial challenges, that there's all these chemicals that are out there. One of them could be good. And then it's estimated there's as many chemicals as there are stars in the universe that we could draw. Carbon gets 4 bonds, nitrogen gets 3 bonds, oxygen gets 2 bonds. If you do the math of all that, we're up around as many stars as there are in the universe. And so, finding that right chemical is a trick, but a lot of people are very invested in this, and they've come a long way. We can make precision medicines today faster than we have ever been able to do this before. Here's another technical vignette where we use this is a robot that's it's kind of like got this Gatling gun of tips that are spitting out here, and I will put some chemicals on this end and then some cells on this end, and I'll mix up all the chemicals. I put catalysts on this plate. I try to make a carbon-nitrogen bond or a carbon-carbon bond. If I make it, they go into the cancer cells and we test. And so that's what's shown here. There's 3 different plates that have data. This is as data centric as I think we're going to get, so just please forgive me. But yellow is good, purple is bad. We made a bunch of chemicals. We mixed them with some cancer cells. Some of them killed some cancer cells, and then we lowered the concentration of the chemicals, and then only a handful of these new chemicals that we had made continued to kill the cancer cells. So this is, there's an enormous space to explore, and it's almost all junk. You find like one little kind of diamond in the rough is the modern drug discovery engine, and that's why drugs are so darn expensive. We get more data, we get lots of data and hone down to this little dot or that little dot. These were all for various types of cancer that we were working on. The point is that we can do this quickly. And so if we can achieve this and imagine a future where we can make a medicine for any health challenge, and you can imagine that AI is a very important part of this discussion, that we've got too much data, can't handle all the data, I can't fit it in my head, but computers get pretty good at massaging that data. And this multi-trillion-dollar industry that is a for-profit industry is quite invested in trying to make things go faster. So it's a very interesting time to be asking questions such as, can we apply this to non-human health challenges? Here's an example. Here's a wiggling protein, and I'm looking at some of the energy interactions that are happening between this potential drug that I would like to make and this protein. And so this is one of the tools that we would use to navigate this. This is actually a protein from an avian flu strain that we had extracted. And it's so what is a wiggling protein? So each one of these is an atom, a carbon or a nitrogen or an oxygen. I've kind of made them covered in a space filling model, so so we don't need to get into those details. And then the yellow bit is the drug that I want to make. And so when you swallow a pill, it's got to get, if it's a pill that you're swallowing, and I'll show you an example where this isn't true, it dissolves in your tummy, it gets across your intestinal barrier into your blood, and then it circulates through the blood. It finds the tissue where the disease is happening, and then somewhere inside that tissue, there's a protein that is driving that disease, or there's more of that protein that's driving that disease. And so that pill is the little yellow. It's this chemical, and that's stopping. That's how we're tamping down the disease. Yeah. What we often do is we'll stick these proteins onto a little magnetic bead. We're going to switch very soon away from technical, so don't worry, just bear with me just a couple more slides. But we attach one of these proteins to one of these magnetic beads, and we'll go fishing for the chemicals that I made inside of that well plate. Beth was with me today giving out little 96 well plates to kids because we're trying to get them started young in this process. So the children of Saba have some 96 well plates that they can start. I mean, we just give them colors like food coloring to play around with, and some little pictures of kitty cats that they can try and make on the well plate. But my hope is that some kid from Saba wants to come and do this type of work eventually. So, okay, so this is one of the proteins that came out of avian flu, and it was the parent clade of avian flu, which had been circulating for the past decade. Was fine, but these new clades, this one came from a bald eagle in Tennessee, was showing resistance to our current arsenal of antiviral drugs. And for me, as a drug hunter, I have my eyes on that. I'm very curious about that. This is showing that when we stick the old school avian flu proteins to an antiviral drug, we're binding to it, but when we get to this new resistant mutant, it's not binding as well. This is a potential problem for us. And we actually got started in avian flu research. We've been applying to the NIH to get funding to do this, and we tell them that we're doing pandemic preparedness, which is absolutely true. We're deeply invested in working on avian flu for pandemic preparedness, but we got started on avian flu 2 years before you were hearing about it at all in cattle and then eventually eggs. I don't know how much of a problem that was down here, but certainly in North America, egg prices skyrocketed because of mass die-offs of chickens. And so now there's a commercial reason to go after this. But my concern was this, that these are the California condors, of which there's about 300 left. And they don't have health care. Actually, they have okay health care, but comparatively, we were very concerned about these creatures, and so we had deployed all of our resources in the days of COVID. As I described, I've enjoyed a career in Alzheimer's and in cancer and many other diseases, and so when we looked at COVID and we thought, well, what can our lab do? We've got this AI-driven drug discovery lab, and potentially an existential threat to humanity in March of 2020. I didn't know, seems terrifying. But coming off the tail end of that, which thank God, we made our way through that. I look at creatures like this and I just think, well, what do they get, right? Like what would we do if there was an outbreak of avian flu in the California condor population? It turns out that all of the California condors have now been vaccinated for avian flu, so other people are working towards this. So there is a path forward. But this, I showed you how we've been thinking about drug discovery. We make drugs. We use AI to make drugs faster, and it really spurred this idea in us that what if we could make drugs so fast, right, that you could make a drug for any disease. And then what if you could do it so fast that you could make it for a disease in a tree or a turtle or a butterfly? That they don't have health care, but we're I hope I'm largely among friends in seeing the value that they provide. And just the need to do that. And so we're trying to launch this idea of conservation chemistry, and again trying to learn from this well-established field of conservation biology, understanding that there is a history of chemistry in the environment that's quite checkered, but we do live in this era where we can create precision health treatments for a lot of different health challenges. And so I hope to talk through just a couple of those. And I'll start with Pebbles the Gila monster, who has kind of become somewhat of a well, anyways, my hero. So we were talking about this at a local zoo, and I was trying to teach my team, who are they came to me because they want to cure cancer, they want to get a job in the pharmaceutical industry. And I said, well, we're going to work on frogs. And so we were having a lot of discussion, and we did our holiday party at a wildlife rescue center. We're talking about these challenges. How could we, this is a couple years ago, how could we take our resources and deploy them in this way? And afterwards, the zookeeper came up to me and said, you guys are talking about all these diseases. This is really fascinating. We have this Gila monster that we're about to euthanize because it's got cryptosporidium, and there's just nothing that we can do for it. And that tugged at my heartstrings, but I'm a pharmacist, and I happen to know that Ozempic and all the other GLP-1 inhibitors or GLP-1 agonists came originally from the saliva of the Gila monster. And so here we've got this multi-billion-dollar pharmaceutical empire, right, that has been built on the biology of this animal. And for Pebbles, it's, sorry, there's really nothing that we can do. So we thought this was a really interesting challenge. Pebbles is really an inspiration to a lot of the local school kids. They like to come and see her. And so we engaged, but we just had no idea how to do any of this. I don't have a background in lizard biology. I've never worked in cryptosporidium, but we were just passionate about this challenge. And so we were lucky to get some poop from Pebbles, and from it we were able to extract the parasite, which I show you here, and we were pretty shocked when we saw it. It's got this this really thick ring of protection around it, and then outside of that, we've been showing this to a lot of cryptosporidium biologists, and they're always surprised by this jelly coating outside of that. So it's like a really protected parasite, and you can't even kill this stuff with bleach. So it's kind of tricky to think of what you're going to do. But that was when. So we got that data here, and so we got engaged here, and you can see that Pebbles' body weight was just kept going down, and then we got engaged right around here. And we had in mind, we were still trying to formulate this conservation chemistry idea, and I'm still trying to formulate it, but we were trying to it's like, okay, what I'm going to do, if I could, what I would have loved to have done was to get some Gila monster intestinal epithelial cells, grown them in a plate, then grown the parasite in the plate, have it infect those, and then test all my drugs against it. We didn't have time for any of that. And so, just in the same way that Michael Soule says that conservation biology is a crisis discipline, and sometimes you need to take actions when you don't have the full data in front of you, we jumped into action. And so my lab is, we also do a lot of work in AI. We do a lot of development of AI. And so this actually came from a high school kid. His name's Tesco Chiganti, and he's an absolute wonderkind. He's helped formulate this whole thing for us. And so he built this up. He said, Tim, look what I did. So no one in the lab had experience in cryptosporidium. Totally new type of disease for us. And what Tesco built was this is the entire medicinal literature around cryptosporidium, and it's just beautiful. So you go to Google and you type in your task of kind of area you want to get into, cryptosporidium, and you just get the first 5 papers that come off of there. So as a scientist, we're often going to the primary literature. We're trying to find these peer-reviewed papers, and what happens is these peer-reviewed papers, they all cite each other. Right, so you get the first 5, and this one talks to like these 20, and then those 20 talk to the other ones, and so he builds out this whole map. So each dot is a single scientific paper, and all that was required was that we would just get 5 of them. You drop 5 in, and it builds out the map of every paper that cited every other paper in the space. And then we live in this modern world with ChatGPT that can read these papers and then give us a map of all the drugs on top of it. And so we went from having no understanding of crypto to kind of like a whole survey of the literature. And from that, we picked out one particular drug called pyromomycin. And I show it here. Now, as a chemist, I could look at this and say, that is not going to get into the blood of Pebbles the Gila monster. I talked about when you swallow a pill, it dissolves in your tummy, it gets across the intestinal barrier, and it gets into your blood. When you have lots of hydrogens like this is not a uniform rule, but normally when you have lots of hydrogens, it doesn't get into the blood as well. And so we thought this would be a good one to stay in the gut, which is where the parasite was. It also so happened by this time the New York Times was covering us, and I really did not want to poison Pebbles the Gila monster while being covered in that way. And so we took this drug, and like I said, what I would have loved to have had was cell cultures of Gila monster cells and the parasite growing on the cell. Didn't have time for any of that. So what we did was we took this and mixed it with more of the parasite that came out of her poop, and got this image. This is a electron micrograph, very expensive way to look at drugs. Everyone I've shown this to, they're like, you screened drugs by electron microscopy? That's that's atypical, but we just, we were grasping at opportunities. And one of the things that we can see is that we have abused this parasite. So if you look at the parasite here, it's got this jelly coating, it's got this thick black ring, it's all dissolving here. Right, and it's like bursting out into these white occlusions inside. So we were really excited. Within days, the Creature Conservancy, who cares for Pebbles, they started her on a course of pyromomycin. They had to play around a little bit with the formulation. And I don't know how well you can see, I apologize for the gruesome image, but it's formulated inside baby quail. That's how Pebbles like to get her medicine. And so, I'm sorry for, I'm running out of, and so we started the treatment, and you can see that her weight was going all the way down. And I'm very happy to report that at the end of the treatment, she tested negative for the disease and stopped regurgitating her food, and she's been very healthily eating quail ever since. So, thank you. Thank you. What's that?
How old is she?
How old? I think she's about 13 years old. Yeah, I mean, this is quite recent, right? I mean, this happened just at the beginning of this year. So, but she's got all kinds of vigor, and so the Creature Conservancy has just been really happy with her health outcomes, and so Pebbles has been a wonderful member of our team, I guess. Yes, question, Beth.
You mentioned the holiday party. Was it last December? Did all this occur
Yeah.
quickly?
Yeah. Yeah, plus everything I'll show you now, which is why I can only stay in Sabo for a couple days. But bless you for bringing me here. So let's do a little trip around the world and look at a few other challenges. I'll start with Florida, although I think it's probably quite relevant here. And so hopefully, this looks familiar to you, so this won't look familiar to you. Or maybe it does. But in Florida, this is the primary cause of stranding of sea turtles. Does this happen here in Saba?
Luckily, no.
No? Okay, okay.
Curacao left and started in St. Kitts.
So what you're looking at, these gruesome images are of a disease called fibropapillomatosis, which I'm very impressed Emily was able to say. And it's a cancer. It's an infectious cancer. It's been on the planet. It's been documented for over 200 years. And, but it's just recently, it's just skyrocketed in Florida. And so we work with the Loggerhead Marine Life Center. They get about 5 turtles a week in this condition. It tends to attack their eyes, and so they'll just be washed on the beach, just, kind of big tumors on their eyes and can't really continue. And so here's Bellatrix and another wiggling protein. So this is Bellatrix, the sea turtle, and you can see she's just riddled in tumors. And so sadly for her, the options that she has available are surgery. But I learned about this disease as I was involved in a startup company called Iambic in La Jolla. We make precision cancer medicines, and so in my one life I was using artificial intelligence and all types of multi-omics, genomics, and transcriptomics, and all the omics to try to find a protein that could be a target of a disease. And then we would go through that organic chemistry bit, design, make, test, make the cure for this type of cancer. We're involved right now with my company in making drugs for brain cancer. It's very difficult to get a drug into the brain. But just having gone through that experience and then seeing an image like this, I'm like, how can we, how can I straddle these two lives where I'm like making this like Star Trek style cancer medicine for patients, and for brain cancer there are very few options. So we desperately need these types of treatments, and we're very invested in maintaining that work. But then I just can't look at a creature like Bellatrix, and I'm so happy to report that the green sea turtle was delisted as endangered just last week. I don't know if you heard this, but it's a least concerned species, and so this is like yet another of our world's major conservation successes. And so I show this that my daughter and I were snorkeling in Aruba 2 years ago. She had just gotten good enough to go swimming. She was 6 years old at the time, and we went snorkeling. It was the very first time she had ever gone snorkeling. We were maybe 100 meters out, and I was like, honey, I think we should go back. We're kind of too far out now. She was 6 years old, first time snorkeling, and she said, I want to see more fish. I said, okay. But then, and just in that moment, a green sea turtle surfaced right next to us, and we just sat there for 10 minutes, just observing. And so finally we swam back to the beach, and my daughter was 6 years old. She says, Daddy, how many times have you seen a sea turtle? I said, well, I think that's only the second time. I saw one in Hawaii one time, and then that's only the second time. And she said, but you went snorkeling lots of times, and I only went snorkeling one time, and I saw one sea turtle. So I think that in my life, I'll see more sea turtles than you. And all right, I'm on board with that. And so I think it just warms my heart that this is. Okay, let's go to the forests of North America, where there's another major challenge. This was actually the one that got me started in the whole thing. That yes, please.
Is ocean temperature a factor?
So there's a good chance ocean temperature has something to do. Of course, pollution should maybe be part. That seems like that's an easy one to point to. Something about their immunity is just weakened, and it tore through Hawaii. It was a major problem in Hawaii, and then it's tamped down. So.
The tumors on the turtles. Right. Sorry.
They thought that the ones in Curacao were mostly impacted by an area where there's U.S. military and putting a lot of ammunition in the water. Heavy metals, yeah.
I think stress has got a major part to it. There's one study that shows that actually the harmful algal blooms that we'll talk about tomorrow, if you want to come and do some toxin testing, that might be a driver. That some of those toxins from harmful algae are oncogenes, so they make cancer take off. It's a really big mystery though. Why these? And it's becoming a really big problem in Florida. just in the past 5-10 years. Like it's really a quite serious problem.
Your
So.
lab is actively working on this?
We are, yeah. I'm just aware of this frontline cancer therapies that are getting incredibly sophisticated, right? I mean, there's still so many cancer challenges that we have today, but if you look back 20, 30 years, the cancer meds we have today compared to 20, 30 years ago, there's a lot in the arsenal. There's hundreds of cancer meds that we have access to. So when I see a sea turtle, it's like, well, maybe we can do something. Do you have a question? Yeah? All right.
I want to hear about hemlock.
All right, so this is the state tree of Pennsylvania. It is the only shade-tolerant conifer that we have in eastern North America. And so when I started native plant gardening during my COVID journey, I wanted to buy this for my garden to put on the shady side of the house. Couldn't buy it. And I tried to understand why can't I buy this in the state of Michigan? So it turns out that there's an invasive insect that the hemlock woolly adelgid that has taken this over. And so when I saw this was at the same time I was working with this company making cancer meds, and we were looking at all types of clinical images of cancer spreading through a body. And so I saw this image of the insect spreading across North America, and I thought that, it just looks like a disease to me. Like there must be something that we might be able to do about it. So this little insect will drop its stylet here into the tree. This little straw goes into the tree. It starts sucking at the juice of the tree and coating itself in this white woolly wax, and then it will explode into 300 eggs, and that egg will travel on the wind and find another hemlock tree, and the cycle repeats 2 times a year. So 2 times a year you get a couple hundred eggs. All the insects are clones of each other, which is kind of funny. They reproduce sexually in Japan, and they need a different tree called a tiger tail spruce to sexually reproduce. But they can asexually reproduce. Alien biology this stuff has. And so what that means is everything that's infecting our the northeastern shore is a clone. And so maybe there's like some like gap in the armor that we can take advantage of there, because they're all clones of each other. But they're such a bizarre creature. And as I talked about, I brought this to bring some chilly vibes to Saba, but you guys are good with chilly vibes here, right? So I won't stay on our snow image. But what I want to show here is that this is our shade-tolerant conifer. So if you know the northeastern United States or Canada, all the trees lose their leaves, right? And so in the winter, all the leaves are on the ground. And what that means is that there's no cover in the forest. And so this tree, because it can live under the canopy of the tree, a pine or a spruce or a juniper, they need full sun. So they can't grow under the canopy of the trees. This is the only conifer that can do it. But you can see the ecosystem service that it provides here, right? This is cover for deer, for birds, and all types of creatures that want somewhere to hide in the forest when all the leaves are down on the ground. So it's a really important species to have available. Now, this has been going on since the 50s. This insect has been there. And so in the early 2000s, there was a citizen science project where distributed through fishing stores and camping stores and the national parks were flyers that said, have you seen a tree that is surrounded by dead trees and is still living? And one was found. Or one cluster of them was found. And this was me last week. It was my great honor to finally visit this site. We've been studying these trees for 5 years. So this tree here is one of the so-called New Jersey bulletproof trees. You can see it's alive and it's surrounded by dead trees. And so through this citizen science effort, they found just a handful of individuals in the Delaware Water Gap of New Jersey. And so you can see here's me with one. This is one on the stream that's just a behemoth, just massive tree, living healthily. And so this is Megan. She's sampling some of these trees into liquid nitrogen, and then we're going to take transcriptomics and metabolomics on all these samples. We've been doing this for a long time. These trees were cloned. You just snip off a branch, put it in soil. You can clone a tree. And those clones were distributed to 8 botanical gardens around the United States. So the Harvard Botanical Garden, we sample every month. There's a research forest in North Carolina, and so it's just a really cool project because it goes back a long, long ways. This is I love the work that I do in our tree space, but what I found is that foresters are on a really long time scale. So they want experiments that, like your grandkid will get the data. Yes.
So, one of the things that you're saying is that when there's a disease that hits all these trees, trying to keep our hemlock alive, that you should not cut them down because one of them.
You're absolutely, yeah, you may have heard, yeah, so thank you for raising that. This is really a key problem that historically a conservation intervention was if something gets in there, go cull the whole forest. And what that means is that we would never find an individual like this one. And so, yeah, thank you for raising that. Particularly New York has been, I think, they've been really on the front edge of that and helping out on that work. And so this, it's taken me a while to formulate all of this, but when I enter one of these conservation challenges, there's both the patient and then there's the disease. And I couldn't formulate it as well today then as I could today. But so this is our study of the patient that we hope that we're involved with USDA and other forestry services who would hope to do reforestation with some of these. And I showed this here. I put this slide in just this afternoon because I thought it might be relevant. The coral community has a lot of similar discussions that maybe there could be winning individuals that then kind of serve as the stock for reef rebuilding. And so I want to get into chemistry a little bit more on this challenge. So here's Dr. Yupu Zhuang, who did a lot of this work with me. He's now a professor in Taiwan. But I show this. We're killing invasive honeysuckle. I love killing invasive plants. And so and what you can see here is that he's got a bingo dabber with some herbicide inside. And so we hack it down and you dab it on there. It still is a herbicide. It's a problem. But it's a very precise application, right? So this is very different than what I showed you of like dumping it at the back of an airplane. We're trying to be precise in our application. This is like our group outreach activity. The tactics for doing this are well known, but what is less well known and what we hope that we can also bring to this challenge is what we know about chemistry. And so I show you here 4 cancer medicines that are all in use. Staurosporine is actually a natural product, and what I show is each of these dots is its ability to inhibit what's called a kinase. So a lot of the biological processes in all of your bodies and in all of biology are linked to phosphorus. You phosphorylate, you stick a phosphate group onto something, and that either makes cancer go up or cancer go down. Phosphorus is like one of the key parts of biology. The kinome, a kinase, is what sticks phosphate onto things. So really, really key thing to be able to do. Nature made a drug for inhibiting kinases, and it inhibits all of them. This is nature saying, do not eat me, or or phosphorus is not on the menu for you anymore. And this is like a wickedly poisonous stuff. It does get used in cancer therapy, but very rarely today. In the old days, they would deploy it, but it's kind of like a pretty nasty chemotherapeutic. And as you can see, this is like a next generation. Here's a somewhat modern one. We're getting better, right? That we can kind of almost take a sniper shot into this biochemical pathways just by changing the chemical structures, right? So if you think back to the wiggling proteins I talked about earlier, and I'm an organic chemist, I want to make this carbon-nitrogen bond, right? I want to install that fluorine just there, and that will teach me, can I hit this with precision? So for cancer drugs, this is very well established. Every pharmaceutical company, I can put this in front of them, they'll know exactly what I'm talking about. You have a question? No? Okay. And so we imagined for the hemlock challenge, what if we could take this principle there? And so one of the things that we're looking at, I've got another wiggling protein, and these adorable bugs, my daughter hates them, but I love them. They're called boogie woogie aphids, aptly named. And so these are in the Michigan forest, and you remember that the bug I'm trying to kill makes a white wax, right? So we were thinking this one is also like a white waxy bug. Maybe there's a selectivity challenge that we need to consider there, and so we've looked at related bugs. It turns out that the protein I've got here, there's no way that we're going to be able to selectively hit this protein in the hemlock woolly adelgid and not also kill the boogie woogie aphids. So it's like the way that we're setting up this challenge, but What we did was we got the genome, or my collaborators in Joe Wegerson's team at the University of Connecticut got the genome of Adelgis tsugae, which is the hemlock woolly adelgid. And we also looked at the genome of these are other adelgids. This one's kind of interesting so at least those of you that are in America or have celebrated Christmas in America, there's a very good chance that you had a balsam fir as your Christmas tree. It's the most popular Christmas tree, and we breed it. It's the most popular Christmas tree, but it's also a critically endangered species in its native environment. So it's like one of these funny ones. There's tons of ones like this. If you go to a pet store and you buy a hamster, it's a Syrian hamster. That species is critically endangered, but it's mass produced. Same with like African violets and all kinds of things. Anyways, I'm on a tangent now. I'm going to bring it back. But this bug is wiping out the balsam fir, which is our preferred Christmas tree. And then honeybees and fruit flies and monarch butterflies. We want to imagine, in the same way I would look at the kinome of kinases and say I want to make a cancer med that hits just one. I want to imagine a molecule that could selectively kill the hemlock woolly adelgid. And what we found was one of them. This is actually a hypertension drug that's being repurposed for this tactic. So with my wiggling gray proteins, we made all those. We can get a score of the energy. What we find is that this hypertension drug very selectively goes after this adelgid, and so we're following up on that research. But trying to take some of the tools that I know about from pharmaceutical research, but then deploy it for rescuing the state tree of Pennsylvania. But it brings me to, okay, so I've had to answer this many times. Okay, chemicals are bad, right? They kill things, and absolutely they do. But so I would argue that they dilute. Okay, bald eagles no longer die from DDT poisoning, and there's a big discussion right now about, because we don't deploy DDT anymore, right? I'm not pro DDT. I just want to make that very clear. But biological tools, when we deploy them, they're in the environment. And so I'm often up against a discussion of we should use the microbiome to solve all of this. And I'm totally on board with microbiota. It's wonderful. You could dab some on a frog or a coral or a sea turtle and it will reproduce. But is it okay if we find some microbiotic samples from, say, Papua New Guinea and then bring them over here to swab on the sea turtles here because it's really good at rescuing their cancer? I don't know, right? But once you make that decision and you deploy it, you can't turn it off, right? Whereas if I go in with a chemical and it's really not having the desired effect, we can stop, and the chemical will dilute on the landscape over time. So all this to say that chemists, I think, have deserved a bad rap in the environmental community, but I would argue that there is room for chemical tools in some of these challenges. And so I'll just close with our work on frogs, which is a major project for us. And I'm going to apologize that the next image is quite gruesome, but I think it's important to see and discuss that we are living in the greatest pandemic our planet has ever had documented right now. It's just that it's for frogs. And so this is the Sierra Nevada yellow-legged frog. They live in about 7 different ponds in and around the Lake Tahoe region, and they're, as you can see, they're in a very bad way. The culprit is a invasive fungus that has emerged, and it just wipes them out. And so we've been working with my partner Tim James, who has developed a really interesting model system based on these African dwarf frogs, which are very easy to breed. They're very susceptible to the disease, and we're understanding a lot about how their immunity spikes, how we can try to tackle this challenge. Of course, we don't like to work in live frogs, so what we tend to do is work with the fungus, which hopefully we'll start here. So it's an ancient type of fungus, and it swims. It swims from frog to frog, very rare for a fungus to swim, but they just kind of, they swim from the skin of one frog onto the next. So we have this tool that I can use, again, in a 96 well plate, like I was playing with Beth, with the kids earlier today. There's none of this fungus in the kits that they were given, just food coloring. But we would use that type of tool to try and hunt for medicines. And so there's one so this one gets deployed. I do a lot of work with the Smithsonian Zoo right now, and this is our status quo. It's called itraconazole. It's a well-established antifungal drug. One of the challenges with it is that it's also an immunosuppressant, and so we give it to the frogs. They clear of the disease, but then their immunity is compromised, and as well, it's quite toxic to frogs. And so, while it is the status quo in frog conservation, I'm the first pharmacist to come into this and say that's not our best drug. I think we could do better. So we're trying to find ones like these ones. So these two here are killing the fungus at incredibly low dose. We're going to be able to dose these hopefully at dozens of times lower dose. And, if you've taken pills, I would prefer to take a tiny little pill than a big horse pill. And so we're hoping that we can go to a lower dose in this particular setting. And so we're doing all of our work in this African dwarf frog species. That will hopefully translate to the Panamanian golden frog, which is kind of our the bar that we are trying to clear. So this animal is now extinct in the wild, but it is the national animal of Panama. If you go to Panama on August 14th, it is Panamanian Golden Frog Day. You'll see all the kids dressed up in yellow, and it's just like a source of so much pride to them. And yet it's not there anymore. It still lives in zoos. Most of the zoos in North America will have living colonies of these species. And a challenge that they have is that when they try to rewild them, they get eaten because they used to be the most poisonous frog on the planet, or tied for the second place of most poisonous frog on the planet. But they have lost their chemical defense in some way through all this process. And so as a chemist, I'm trying to come in and help them recover their toxin. And that involves a lot of screening streams and trying to find some of the precursors to those toxins. So I hope that some of you will want to join and learn a lot more about this process tomorrow. But we're hoping that we can, in the same way that in the pharmaceutical industry, I've spent a career working in test tubes, then in a mouse, and then in a rat, and then maybe in a dog, and then into a human. We do this translation. What we're trying to build is we're replacing human with Panamanian golden frog, and then working with increasingly easier to work with not endangered organisms. This is the lemur leaf frog, which is another absolutely adorable one, as you can see. Yeah, Larry.
So the Panamanian yellow frog, there must be something in its natural environment that provides the toxicity which they don't get in the lab.
That's our hypothesis. It's either in their diet or it's in a microbiome strain that they used to be able to make it. But it's one of these ones that has a lot of lore that they were used in tribal rituals and so so it's really interesting. A very interesting challenge. I've had opportunity to deploy some of our work in Panama, and so these are critically endangered. These are Atelopus varius, and so this was earlier this year. Was a big career moment for us that we were able to work with the Smithsonian Zoo on developing a tool that's helping them with their frog husbandry. And so this is a critically endangered organism that's getting a precision treatment. And as I described, the Panamanian golden frog is kind of one of our main focal points, and so I invite you to come and explore this more tomorrow. Yeah.
How do you distribute the treatment? Is it individual or?
At this point, yeah, because everything is individual. Like I showed you those the yellow-legged frogs, that really gross picture with the frogs. They actually live in 7 ponds, and teams will go to those 7 ponds every year and collect all of the frogs and treat them. So if you think about like these endangered species challenges, it seems too daunting. Like how could you ever do something on an environmental scale? But in reality, a lot of them are in pretty localized environments. And then most of the work that I've shown thus far is in a captive setting. The state tree of Pennsylvania, they actually do treat almost the entire North American seaboard. Some states invest more in it than others, and they'll hammer an insecticide into each individual tree. It's a pretty large scale effort. Yeah. Thanks for the question. And so I think, yeah, I'm at the end now. So just to say that right now in my lab, we have many projects, but there's 3 major projects where we're actually trying to invent new precision health treatments for avian flu with an eye towards conservation for the state tree of Pennsylvania and towards frogs. And so while we also enjoy all of the drug discovery research that we do for classic medicine or modern precision medicine, we try to leverage those tools and point them in this new direction. And so I'll just close by saying that AI is accelerating drug discovery. Hopefully that is something that you can agree. Hopefully I've convinced you that chemists can be conservationists. This is a big part of my mission, and one of the tools that we have, or one of the reasons that we've got something unique to bring to the table, while there has been a long history of chemicals having a decimating impact on the environment, I argue that chemicals will dilute and you can turn it off. So it's a conservation solution that you can at least stop if it's not going well. When we introduced cane toads to Australia to deal with the beetles that they were meant to deal with there, that turned out not to be a good conservation intervention, right? And there's still cane toads in Australia. And so, a chemical tool, at least you've got the opportunity to turn it off if it's not working for you. Cheer me on as we try to launch this field. I thank these guys, and thank you all. Yeah.
How much red tape do you have to go through with the federal drug people?
Well, so the first part of your question,
Dealing with drugs?
I'll repeat the question. So the question was, how much red tape do I need to go through with federal drug, with the FDA? So with them, we actually haven't approached the FDA. So we haven't gotten that far yet. We go through a lot of red tape with U.S. and Fish and Wildlife that steward these endangered species. So there's a lot of work that needs to be done. I didn't bring it into this, but we have a lot of discussions with communities and ethicists to try and understand what's the appropriate way to engage in this. What you're alluding to is, or what I believe, like, to have a conservation tool, it needs to be approved by somebody, but it actually probably won't be FDA, totally depends on the species. If it's like a sea turtle on land, that's different than a sea turtle in the ocean. If it's a tree, that's different but so largely it's the agricultural entities that will oversee this, or Fish and Wildlife.
And they've been more amenable?
Well, I didn't say that they've been more amenable, but yeah, I think we haven't yet had a chance to register a new product for this purpose. So I can't really say. I can say that we sometimes spend years getting the permits just to do the work.
We know that
Yeah.
with the drugs.
Yeah. Oh, well, then actually launching. I think, another challenge is just who's going to pay for it. And my dream is that it could be a nonprofit, that there could be. What if there was a nonprofit that went through that whole process and then deployed? To her question, these are pretty small scale, right? We're not talking about deploying to massive populations. There's just
You hope it's going to grow.
right.
And is there anybody in Europe doing the same sort of work?
So I'm unaware of a chemist who's doing this type of work. There are incredible conservation stories happening in Europe with frogs, for instance. There's the Majorcan midwife toad, which lives in one single pond on a mountain in Majorca, and they helicopter in every year, and they treat all of the frogs that still live there. They scrub all the rocks and disinfect them and put them back just to keep the species alive.
But what about the ash dieback?
The ash borer.
Yeah.
Yeah, that's another major one. So, and particularly where I come from in the Midwest of America. So there's a beetle called the emerald ash borer, which has just wiped out the ash trees. And so that one, I'm not engaged, but our partners, when I talked about trees, are deeply involved in ash. And that has been a very successful program in terms of identifying these lingering individuals, where I showed that we found these trees. And so ash has actually had more success in that space. Right, right, right.
Someone had better work on the lanternflies because they are invincible.
Right,
And they can like travel on seas. I mean, they're really incredible bugs. You know, so look very beautiful. But
So the spotted lanternfly is a recently introduced invader to the northeast of America, and they're absolutely beautiful. But you should kill them. Yeah. I mean, that one we haven't engaged in that fight yet, and I find that one is really I find that one is tricky because they spread so fast. They just spread so fast.
they spread so fast, they're extraordinary in terms of their abilities. So,
Yeah.
How much are you thinking about, like the things we talked about today, how much of that was, is about prevention as opposed to treatment? So like preventing them from getting it.
Yeah.
Or getting it once they have it.
You know, I ultimately didn't put in slides, but so I think when that What is Conservation Biology paper came out, this wasn't really well understood, what drives extinction. But today we have a pretty good understanding that there's 6 main drivers of extinction. So what I'm interpreting in your question is why not climate change, why not habitat loss, and why not over harvesting? Those are other major drivers, and so those are definitely 3 of the main drivers. The other ones are pollution, chemist, right? And invasive species and disease. And so we focus on invasive species and disease, but I think that the conservation chemistry idea kind of captures all of it. Like I have a lot of friends who do environmental analysis looking for pollutants, and I would say, well, that's a chemistry challenge. But so I think I'm coming in looking at disease, which is kind of putting a Band-Aid on the bigger problem, right?
And that's treatment as opposed to vaccination.
Oh, I mean, I guess I'm pro-vaccine, I guess, or well, don't put that online, I guess. But yeah, I mean, I'm okay with the vaccination strategy. I think that. So the nice thing about a vaccination strategy is that you're kind of one and done, right? I mean, my friends who study infectious disease always describe that what you want in an infectious disease challenge is both a vaccine and a medicine to deal with the patients that you just couldn't vaccinate.
My question goes back to vaccine and the condors. So how big of a concern was it, and how much red tape did it take to vaccine the last 300 surviving birds and making sure that you weren't killing them?
Yeah.
Causing another problem.
So, I mean, it's interesting. The company that provided that vaccine makes chicken vaccines, right? So they had a lot of experience and understanding of how this works. And then the other thing that I think is really interesting there is that there's like 300, I think maybe 328 or something like that condors. We know all of them, right? They all have a name, they all have a tag. We know exactly where they are. So it's kind of, I mean, that's not the case with a lot of things, but the California condor has really enjoyed this, or shouldn't say enjoyed, but it's been studied as a conservation symbol for a long time, and there's kind of a lot that is known. So while it seems absolutely daunting, I think that it's possible in that setting. And then, so I don't know what was done in terms of preparing for that. I know that U.S. Fish and Wildlife were very involved. They talked about it on their social media. So they're involved, and the U.S. National Parks, I think, were also involved. But when we were exploring antiviral drugs conceptually for condor, what that would look like, and the teams that we were talking to were proposing that you should study vultures because they're not endangered. Certain species are not endangered, but they're quite close to condors, so you could get some better understanding than chicken, right? We've got a lot of data on chicken drugs.
You seem to be having a massive bird flu epidemic through the turkey chicken.
Yes. Yeah. Yeah. So that, I mean. Right. So I mean, yeah. So we, as I mentioned, we got involved in it through the lens of conservation, long before that was, that you were hearing about these kind of mass die-offs in chicken farms. I mean, the kind of the wild creature or even captive endangered organism is quite different than a broiler chicken that, you know, is like sitting on top of another broiler chicken. So I think they're just kind of, the opportunity for disease spread in that, in commercial farming is quite, right.
It has a big economic.
It absolutely does. Yeah, absolutely. And so I think it's interesting when you can think about the economic drivers. I like to think about like Pebble, the Gila monster, who, this creature, we've sold, we haven't, but like the many billions of dollars of these drugs have been sold, and they just wouldn't exist if it wasn't for that animal. So I think it's interesting. Thank you. Thank you so much.
Thank you everyone for coming out tonight. Thank you, Tim, for an amazing presentation. And if you're interested in joining him tomorrow at 3 o'clock, please see Lynn or me. And everyone have a good night.
Yay. Thank you guys.